Thanks, Nikki. And thanks to all of you for sticking around right before lunch. Okay, so this is all about research that is focusing on what can we do at early life stages to intervene to help corals survive future climate change, but with the idea of scalability. And this is a photo of a reef in Kaneohe Bay, and our two main reef-building species are Parades compressa in the back right there, and Montipora capitata in the front. And the first part of this talk, I'm going to focus on this question of, will thermally conditioning larvae help spats survive a heat pulse? And the answer is no. And so this is a paper that came out this year where we talk about it. And what we did is we had larvae, we exposed them to a cool temperature, an ambient temperature, and a hot temperature. Put those larvae, settled them in ambient water, and then put representatives from each group into an ambient tank or a high tank to see if that pulse at the beginning of their life had any impact on their survival over time. And the answer was not really. So on the x-axis, we have the days of that exposure, of that heat treatment as spat. On the y-axis, we have percent survivorship. And you can see at the top, the ambient temperature, there was really no significant differences between any of the preconditioned treatment, which is what we expected. But then at that high temperature, we had significant die-off. And so for this particular species, this type of thermal conditioning did not have an impact. What I wanted to spend the rest of my 4 minutes on with you today is a lot of work during my PhD that did not work. And this is this whole idea of can we manipulate the coral algal symbiosis to increase thermal tolerance? And this is very broadly a yes but kind of question. And there are two kind of things you need to know to sort of understand why I did what I did and why it didn't work. Is that we have different types, different species of algal symbionts. Those have different thermal tolerances, and we find that they can confer those tolerances to corals. The second thing is how corals get their symbionts. So there's two main methods. We have vertical transmitters, which are packaging up those symbionts from the parent colony and then giving them to their offspring, so the larvae are born with symbionts. And this is the case for the majority of the species that we see in Kaneohe Bay, which I know is not the case for all of you out here. And then we have this horizontal way where they're born without symbionts. They get them from the environment and things around them. So trying to understand if we can do this and if it would work, there's all these steps. Remove the symbionts that are there, inoculate them with new symbionts. Will they take them up? Will they establish a population? Will that even matter for thermal tolerance? And what I spent the first 3 summers of my PhD trying to do is just get the symbionts out. And this was really hard to do. I tried this at all the life stages, but I'm going to focus right now on the larval and the spat stage. So we tried a lot of temperature treatments. You'll see a lot of plots like this. It's on the x-axis we have time. On the y-axis we have percent survivorship. And we first tried raising the temperature. Kaneohe Bay usually beaches at 31, so we knocked it up to 31 to 32. And what we found is that within about 2 weeks, everybody was dying off. We had only about 25% survivorship, and then pushing a month, it was even lower than that. And what I was doing during this time was taking larvae from all of these treatments, squashing them under the microscope, and then just looking, are there symbionts here or not? I have a lot of samples that I never bothered to count because really the question was can we get rid of all of them? And what I did is at this particular time point at day 20, on average, corals from any of these treatments had more than 1,000 cells per larvae, so this was not going to work. Then we went to the next month of spawning and decided to try something else. So I went cold this time, that first experiment, see maybe that would help. But then we added a light treatment, so a high light treatment at ambient temperatures. And what we found, similarly, everything died off pretty heavily by the second week. But at day 5, when I counted all those symbionts, the light treatment really stood out. We had a significant drop in that symbionts per larvae. And these are larvae that look good, not the 3-pronged kind of unhappy-looking larvae. These are larvae that we might consider using for experiments. However, it wasn't until day 39, and I remember this so well, where I was sitting there at the microscope and I found the first larvae that looked normal that had no symbionts. This is way after survivorship dropped basically to 0. So when we talk about scalability, I think biologically, yes, it's probably very possible that we can do this, but it's not scalable for this species in this particular way. And that was our larvae are very fragile. We're not necessarily expecting them to bleach out. So we did a lot of experiments like this also on freshly settled spat. And the temperature treatments didn't work. That's sort of the spoiler. But I wanted to just highlight, we used the Matthews protocol using menthol to chemically bleach the larvae, because our thought was that if we could use chemicals, bleach them faster, it might make them happier. We were able to bleach spat entirely, but most of them sort of looked a little unhappy like this. We saw a lot of the tissue starting to pull back. We were feeding them heterotrophically to keep them happy. And then we also tried some inoculations with these corals to see if they would take things up. And we did this in a couple different ways. So I ran 2 trials. First, as soon as we found corals that had gotten rid of all of their symbionts, I was like, all right, let's get these symbionts in there. We just squirted them in the water. I tried to squirt them right into the oral pore. We fed them to their food, see if they would stimulate feeding. Would they take the symbionts up then? The answer is not exactly. And then I talked to some collaborators who found that they had to actually wait about a month for the coral post-bleaching to kind of regain its composure to then decide it was okay to take up these symbionts. But we didn't see this happen. So C15 and Kaneohe Bay are the ones that were the more thermally tolerant Cs that we had, and that's what we were interested in. But just to make sure it wasn't me necessarily, I also tried to infect them with their own symbionts, which are C31s. And we do have one horizontal transmitting species in the bay, Lobactis sutaria. I raised those, settled those, fed them their own symbionts, fed them the C15s and the C31s, which we were isolating fresh for each of these experiments. And these corals took them up, as we've seen from previous research, just to sort of make sure it wasn't the freshly isolated symbionts or any sort of methodological problem we were having. And so in that sense, for vertical transmitters, this was a huge roadblock, getting the symbionts out. We did try this on adults as well, and also ran into a lot of the same problems. But basically to see, can we really answer this question, I went to Australia, collaborated with folks in Madeline Van Oppen's group, and we were able to successfully do this with horizontal transmitters in larvae, where we inoculated them with symbionts that they're naturally found with, and then symbionts that are usually found in sponges or forams that have higher thermal tolerances. And we did see evidence not only that they would take these symbionts up, but that there was higher survivorship in the larvae that were inoculated with the more thermally tolerant symbionts. So I think that is the end of my time. Thanks to all of the people who helped me, collaborators. This was all sort of a brainchild of Ruth and Madeline's Paul GL and Family Foundation work. And if there's time, I'll take any questions. Thanks.